right, thank you everybody. I want to take you on a journey into a fascinating world that has more to offer than what meets the eye. When we look out on the ocean, all we see is like a blue surface. Whenever we want to look beyond the rippled blue, it's kind of difficult. Like our bodies are not really equipped with the right sensors to explore the underwater world. So underwater we can't breathe, we can't see very well, we can't hear very well, our sense of smell is completely gone. So we need some kind of technology to explore the underwater world. And similarly in oceanic exploration, we need technology as well to find out what's happening in the ocean. And in modern oceanography, these are normally very fancy, sophisticated technologies that produce very vast amounts of data. And we need a bunch of scientists from different fields to first process the data, analyze the data, visualize it, and interpret it as well to make some sense of it. And one of these person, well, that's me. And now let's dive straight into it. Please meet Zooglider. Zooglider is a 2-meter-long orange robot with some wings. It was developed by the Scripps Institution of Oceanography in San Diego, California. And Zooglider can dive down to 400 meters, and it does so without the need of any engine. It just has a small bladder, and oil is kind of moved in or out of this small bladder, changing the buoyancy, making it dive down or come up to the surface again. Zooglider was specifically designed to observe zooplankton in their natural environment in a non-invasive and quiet way. And like many of the organisms Zooglider tries to observe, it uses like acoustic and optical sensors. It also has sensors for temperature, salinity, pressure, or depth, and it also senses the phytoplankton that surrounds it. Zooglider is also kind of smart, so it can detect the seafloor, so we can send it out into complex environments like underwater canyons that are very difficult to sample otherwise. Now, let's first have a look at the optical sense of Zooglider. So Zooglider has this sampling tray permanently attached to his nose. And we're talking about zooplankton that we want to observe, and they are just drifting around. So when Zooglider ascends to the surface, loads of zooplankton is passing through this sampling tray. There's a red LED light that shines light. On the other side, there's a camera. Everything that passes through, even transparent and translucent organisms, will cast a shadow. We record the shadow, and we get these beautiful, elegant silhouette plots or shadow graph images. And this technology allowed us already to get new insights into like the physiology of some organisms, like these beautiful ctenophores in the corner, these very fine tentacles that we were unaware of. We got new insights into behavior and into prey-predator interactions that we couldn't observe previously. Now, optics are great for like differentiation and identification, but there's loads of limitations because optics only reach so far. Light doesn't penetrate very far underwater, and in this case we also have a very limited sampling tray, but marine organisms also want to see far, but light is kind of like, it dissipates very quickly underwater. So if we just move down 10 meters from the surface, 50% of the light is available. Once we reach 100 meters, we're in complete twilight zone, and anything below that is eternal darkness. So that means over 80% of the oceans is just eternally dark. So that's a problem. But where light vanishes, sound prevails. So underwater, sound travels about 4 times faster than it does in air. So that makes it quite a good tool for like orientation and also for communication. So let's look at dolphins, for example. Everybody knows dolphins use echolocation or clicking sounds to orientate themselves so they can navigate around, they can detect friends, they can detect enemies, they can detect prey, they can change the frequency, so they use a low frequency if they want to look far away, they change to a higher frequency if they want to inspect it a little bit closer, get a bit of a detailed view. And now we have learned from our animal friends, and we equipped Zooglider with a kind of like echolocation tool as well. So we gave it an echo sounder. So Zooglider can now send out sound at 2 different frequencies and detect what's happening in the environment. So like this, we get a much bigger sampling volume than we had with the optics. Now, how does it work? Basically, sound is just a pressure wave. So sound travels through the water column, and whenever it hits a target with a density or sound speed that is different from the surrounding water, a part of that energy will bounce back or echo back to the source where it came from. Now, we can measure this echo, we can time it, so we know how far the target is away. We analyze the structure and the amplitude of the signal. Then we know like a little bit about the material properties of it. We know a little bit about the size of it. If we use one frequency, it's a little bit like watching black and white TV. So we see like contours, we see shapes, but we miss some of the details about like the internal structure, the structure of the material that we're looking at. So now if we add 2 or multiple frequencies, we get more insights into the structure and material properties of what we're looking at as well. So that's what we're looking at here. This is just a full mission of Zooglider in terms of acoustics. So we have 2 frequencies that we differentiated. And whenever you see something in dark red, that's more small organisms dominating. If you see something getting lighter white or blue, that's more larger organisms dominating the scene. Yes, and with this technology, we already managed to detect that zooplankton of larger size migrate deeper and faster every day than smaller organisms, and this has big implications for the carbon punk and the carbon sequestration that we're doing, and has impacts on what happens with climate change to these organisms. Okay, and then if we combine the acoustics with some other biological samples, be it from nets, be it from optics, or nowadays eDNA, we can get the best of both worlds, and we can differentiate in high detail and get like a high sampling volume as well. But normally we have acoustics, they're not mounted on fancy equipment like Zoo Glider, but we have them mounted on small to very large vessels or research vessels. And the example I want to show you here is collected by some friends at the Australian Antarctic Division, and this is some data we're actually analyzing right now. So what we encountered is like we came across what we call a mega swarm in Antarctica. So this is a 9-kilometer long krill swarm that we observed. And what we're looking at here is a cross-section of the swarm. So we have the 9-kilometer long swarm, and the boat went over it like this. So that's what we're looking at here from left to right. And then on top, we have the surface waters, and the bottom of this plot is 150 meters of depth. So this krill swarm in the middle, all this green blob kind of thing, that's all krill. The cross-section is about 100 meters in height at this location. Then on the surface, we see some noise that is caused by the vessel. Then we have these vertical, like horizontal marks, and these are caused by whales that passed through the krill swarm. They were eating a little bit, scared the krill away, they created a gap in our data. We have this very beautiful vertical mark, so that's a full attack of a whale. Whales dive down deep to feed on those, and the krill disappears because they had a bit of lunch. And then we have this very beautiful spot in this echogram. This yellow blob that you're seeing, that's a whale just eating krill right now while we were observing it. So this is kind of like a fascinating thing to look at. This allows us to have a non-invasive way that gives us a full view of the water column. We can observe these prey-predator interactions in real time without disturbing the scene. And I think that's kind of a beautiful thing to do. Now, moving from all the way south, let's move all the way north. Now, picture the scene. We're sitting at the beautiful Norwegian fjord. The water is calm. We're enjoying the serenity. And then there's some scientists coming, and they want to know what's happening underneath the surface. So we dump in an echo sounder to see what's happening below. And up to now, we spoke about single frequencies or multiple frequencies. So we say we send out a low frequency sound, like boom, 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 or a high frequency sound, bing, bing, bing. But we can also modulate the frequency. So we can go from low to high, make a chirp, make a frequency modulated sound, like boop, boop. And then you don't just have like single frequencies to look at, but we have the full spectrum that we can analyze, and we get a much more defined view. We can resolve targets at a much higher detail. So that's what we're looking at here. What is this abstract painting showing us? We have these horizontal lines, and these are all small fishes. In this case, they are pearlsites, Maurodicus mulleri. Pearlsites are some very beautiful silver fish with beautiful light organs on the belly. But they're not just beautiful, they're also hungry fish. And what are they doing? They want to eat copepods. Copepods are a kind of zooplankton species with an oil sac, so they're very, very nutritious, and the pearlsites want to eat them. Now, I said before that zooplankton are just drifting, so what are these weird zigzag shapes that we are seeing here? And that's kind of fascinating, because you have to imagine these copepods are just like a couple of millimeters in size at best. And now we see all these traces on the echograms. And that's because copepods have this special technique. They can, if they feel scared, they can do this burst. And they're actually the fastest animals on planet, relative to size, of course. And they can make jumps of up to 800 millimeters per second. If we translate that to human size, that would be roughly 6,000 kilometers per hour. So that's quite a fast sprint. Now again, I think this is extremely fascinating. We can observe these prey-predator interactions in real time at a very, very fine scale. And if you would have asked me just a few years back if we could ever resolve the escape movement of copepods by just using acoustics, I would have said, no, that's not possible. That's way too fine a resolution. So I find this quite fascinating. And of course, it tells us what we might feel like that's a beautiful serenity. Copepods feel it's kind of a stressful world. Now, taking a step back, generally we don't do these fancy experiments with our acoustics, but generally what we do is we go out on our research vessel, we follow predefined tracks, we record all the echo that we attribute to fish. We take that echo, we divide it by the known echo energy of a single fish, and then we can translate this into biological meaningful things like abundance or biomass estimates. And this is one of the very important metrics to go into like a scientific stock assessment and scientific advice of how many fish we can sustainably catch or might leave in the ocean. And again, this is just to give you an idea what this data can look like. When we use broadband technologies, we can see individual boomerang tracks there. All of these are individual fish. So in this case, we don't just see a blob, but we can see all the individual traces, and we could even go ahead and count the fish individually. We can measure how fast they're swimming, which direction they are swimming. We can estimate their size, and with the broadband technology, we get much better differentiation capabilities as well, and we might further reduce the need for direct biological sampling, so make it a less invasive technique to monitor our fish stocks. Now, we talked a lot about getting details using one frequency, multiple frequency, broadband technologies, but what if we want to know more about the full 3D structure of these fish schools? Now, a few years back, I went out with my colleagues from the University of New South Wales, and we were happily surveying along the coast of eastern Australia. And then we came across this beautiful school of Australian mackerel. And we had our multi-beam echo sound on board, so we just saw in real time this full 3D structure of the swarm. And then what we can do is we can see the full internal density of this fish. So how does it change from the center of the school to the outside of the schools? We can see the individual fish swimming above, below, and around the schools. So we get new insights into the behavior of fish. We get a better error estimate of like how the fish are distributed and how the abundance of the fish are in the area we want to observe. So that gives us quite a lot of new insight and information that we can get in real time. Okay, and now we talked a lot about biology. Let's go to the landscape. Now, one thing that I find quite weird or bizarre is that we know more about the surface of the moon, the surface of Mars, than we know about our own seafloor. Still, over 70% of the seafloors remain like unmapped. But luckily, multi-beam sonars are changing this quite rapidly. And what we're seeing here is just an example that was collected by my colleagues at CSIRO in Australia. And just a few kilometers off Tasmania, so not even far away, they discovered this underwater volcano range that is with mountains that are up to 3,000 meters in height, and we didn't know anything about this. And just 2 years ago, some friends, they collected data around Antarctica, and they discovered a 20,000 square kilometer mountain range that we didn't know about. And even I myself, when we were sampling around Mertz Glacier in Antarctica, I stumbled upon an underwater canyon that was uncharted. So that's just to give you an idea that there's still a lot of things to find out and explore in the world outside. To take things even further, we now develop new technologies, and this is an example of a synthetic aperture sonar. So that's in simple words, it's just like instead of taking one ping, we take multiple pings and we all combine them together to get a better resolved image, and now we can resolve the seafloor at the centimeter range. So what you're looking at here is this rack next to this little hill, and this is purely based on acoustic data, so there's no imagery involved. And I think it's kind of nice if you can get like almost photographic images by just using sound. And now, one thing that is very important to me is up to recently, acoustics was a domain that was very limited to big research institutes. We needed like a lot of expertise, expensive software, expensive technologies, but nowadays we're working very hard to develop open source software, AI models that are easy to use, and open source or cheaper hardware as well to make this a more democratic part of science. And now to conclude, I want to show you this picture of an artwork by Claudine Arendt that uses the acoustic response of zooplankton as it is recorded by Zooglider and transforms it into porcelain vessels for all of us to experience. And this is where science and art really meet. It's like we try to take the invisible or take the complex world and transform it into something that we can understand or that becomes tangible. And so next time you look out into the ocean, remember there's more to it than what meets the eye, and you should just listen a bit more closely, and we might just understand a little bit more about our ocean, about our planet, and maybe even about ourselves. Yeah. Thank you.